Hi everyone, thanks so much for coming today. Really excited to be doing another session of quick fire talks. So we're going to have 4 speakers, starting with Peter, and then we have Faye, and then Emilio is third, and then finishing off with Beverley. So we're going to have a range of different topics. I've said nominally 10 minutes, but I'll keep an eye on the time. If anyone's going over, I'll probably start like waving at you. But yeah, I think we should be pretty okay with time, and we might take 1 or 2 quick questions between each speaker as well. And of course, if there's spare time at the end, then we can do more questions or chat as we like. So first of all, we're going to have Peter. So everyone make him welcome while I do the slides. And then you know how to drive. Can you put that on, please?
Good afternoon, everybody. I'm Peter Smith, Senior Research Engineer with the Olivastri team, and today I'm going to present about a project that you may not have heard about with a lot of collaborators from the centre. It is about green classification of both turtles and birds. So here in Australia, we've got the Great Barrier Reef, and we have these nice sandy islands. And during the breeding season, we get a lot of turtles and a lot of birds that come around. And it's great if we can accurately and consistently count how many there are. It's good for conservation, and we want to do this using automation, so that it's easy. So I'll start off with the turtles. The way that they do this is pretty ingenious. There's a bit of a trouble. Although you have thousands of turtles showing up, they usually are only at their resting sites at night, so they're only on land during night, and the drones that we fly over to create the geotips can't see them. So what we do instead is we have a shore party that goes onto the islands at night, and they have like a big bucket of paint. This is non-toxic paint, and it washes off in a week, so no harm to the turtles. And they paint a set number of turtles. Then during the day, as they disperse, we can count the ratio of turtles. And so this is what it ends up looking like when we run the inference. As you can see, it's picking up turtles, and then it's classifying them as either painted or non-painted. So using this ratio, we can get an idea of how many turtles there are. The pipeline looks like this. There's 3 steps, and kudos to Dorian, Java, and Serena on setting this initial pipeline up. So you have the 3 stages. First, detects the turtles, it then tracks them, and then it needs to classify them. This initial version was done with YOLO v5, and the way it worked is we took the videos and we scaled them down and ran the inference. This worked really well in the open water. However, when we get to the reef, so you've got these large sandy islands, some of it is surrounded by nice open water, which is great for our pipeline, but also there's a lot of reef, and that is a trouble for our pipeline as well. So some of the latest work has been to improve the inference. And to do this, we wanted to increase the resolution. So kudos to Matt, Alan, and Serena for setting up Sahi. It's got a bit of a... Whoa, okay. Turn off the audio. Anyway, so it splits it up into different segments and runs inference on each of those segments. And then we combine them later. This allowed for more than double the resolution for the turtle classification and really helped to make the pipeline more robust. All right, I will now talk a little bit about birds. So this was taken, this was done using a data set from some ornithologists, so experts in the bird field. Obviously, I don't know too much about the bird field, and we got them to, they had already had this amazing data set of the classification of the birds. And so there was a collaboration between Monash University, where the ornithologists are, and Serena to take this data set and use it for machine vision. Matt Tab, then in version 2, set up this automated pipeline. While we had the inference model, we had these massive geotiffs that would span the entire island, and we needed an automated way to provide a classification count of all of the birds. The pipeline, fairly simple, but we take the geotiff, which is created from the lawnmower pattern that the drone takes, or that flies over the island. We split it into two parts. So the downscaled part, which is for the UI, and I'll talk a little bit more about the UI a bit later. And then we have a full resolution, but in a new format that we can use for splitting up. We're going to split it up, classify each tile, merge, and we're going to have some overlapping classifications during the merge. So then the last step is to detect the duplicates and make sure there's only one in the count. And finally, we output the results in a UI-friendly manner. Next, I want to talk a little bit about some of the recent improvements. Some of them we got for free. So we've been using YOLO, and over the years, they've had a whole bunch of new releases. We recently upgraded to YOLO 26, but we're also still using YOLO 11. We started way down at YOLO 5, so just by upgrading our libraries, that's a free win. You do have to retrain the models, but really helps. And I wanted to include this a little bit here. When I uploaded the images to Microsoft PowerPoint, Copilot helpfully tries to classify them for you. And Copilot's like, oh, this is a group of fish swimming in sand. And I'm like, okay, thanks, Copilot. But it really hammers home why you need to make these specific custom models to get a robust classification. Matt Tab helpfully listed a bunch of challenges that I can talk about here. So it's challenging to get a good label of these birds because of varying lighting conditions and different just variations in the islands that we run these models on. Also, flying birds are a challenge. They appear different enough that the classification model gets a bit confused, so we need to split them out first and either discard them from the count or run a different inference. Also, there's enough visual difference between offshore and onshore birds that we need to have good training examples of each. And finally, we couldn't cut any corners with the classification. We needed the color, so we can't just run the classification on a black and white. Next, I want to talk about how we tied it all together. So this was Alec and myself. What we did is we had three separate components. We have the turtle finder, the bird finder, and the UI. And we decided that the ROS communication framework was the best way to link them together. And Alec and I worked on building out a UI that was both user-friendly and intuitive, so that we could hand it off to non-technical users and they can just take either the geotiffs or the videos and get a robust count from them. We're now at version 3 and we're pretty happy with how it's going. Here's a big list of all the collaborators. It was a collaboration between not only QT but also Monash University. We've had the Australian government helping out with, and we've had members from both QCR and the ref team helping out. So I think I sped through it pretty quick, so open to questions.
Well done.
Thanks.
Any questions for Peter? I had one. So the end users for this UI, is that mainly rangers, or is it government, or is it scientists, or who's the person actually using this?
We have an end user in Ames that they take these videos and these geotiffs and they do some conservation work, some calculations,
I come along and want some other object of interest, so other than turtles and birds, Is it just training the classifier and everything else in the pipeline remains the same, or like how much is reusable?
Yeah, actually, as long as whatever you want to classify can either be done with a similar pipeline, as in if you had some kind of GeoTIFF and you wanted to classify everything of a type within it, you could use the burn finder. And if you had videos, then you could use the turtle finder, even though you're not using turtles. Anything that you need a different pipeline would be more work. But it loads up the models, it reads the different classes, and you can select which classes you want to identify, which has been useful for some of the islands. We know specific bird species don't exist or don't roost there. So we exclude them from the classification as well to get a more robust solution. So yeah, it is.
Awesome. We might leave it there so we can keep rolling, but thanks again, Peter. Full screen.
Like default the most
What have you done? Control F5.
Yeah.
Oh, yeah. Okay.
I can make it small.
Yes.
All right, all good. Beautiful, thank you. All right, hello everybody. For those of you I haven't had a chance to meet, my name is Faye. I'm a coral reef ecologist, fairly new to the lab, where I've just started my PhD working with Scarlett and Toby here and with Nikki Brown from UQ. Coming from a non-engineering background, I want to kind of give you a whistle-stop tour into coral reef ecology, what we monitor, how we monitor, why we should care about monitoring, and particularly the role of AI now and into the future. That's not working. There we go. So briefly, I'll get everybody up to speed on what corals actually are, why they're important for us all, how we monitor them from an ecological perspective, how we collect our data, what we measure, how we're using AI now, and what some good directions are for the future in AI, particularly in coral reef ecology. So as some of you are already hopefully familiar with, corals are sessile animals, meaning that they don't move. They live on the seafloor. They have hard skeletons, and over hundreds of thousands of years, they build reef structures together. They come in many different colors and shapes. Their color is given by their symbiotic relationship with an algae called zooxanthellae, which gives them the different appearances. And the most common shapes that we find are encrusting, tabular, and branching corals, as you can see on the bottom. All of them are adapted to slightly different environmental conditions, such as light penetration, wave action, and so forth. And they're characteristic of different zones along a reef. So we find them in different spaces. Coral reefs provide an array of ecosystem services upon all of us in the room also rely, such as protection from storms, tourism, sustenance of livelihoods, and so forth. And even though they cover only 0.2% of the seafloor, they provide habitats for nurseries, array of services for up to a quarter of all marine animals in the world. So this is a direct motivation as to why we want to monitor them. To make it a little bit more specific for us, bring it closer to home, in Queensland alone, we have over 400 types of species of coral. But at the same time, unfortunately, corals are the second most threatened species set in the world. Like any animal, they're very sensitive to the environmental ranges that they are accustomed to, particularly temperature, wave action, light, and substrate, so the hard bottom that they can grow from. In addition to this, all of these things are changing in relation to global and local stressors, most predominantly global warming, so rising sea surface temperatures, but also pollution, overfishing, and mechanical damage from tourism and boats. Monitoring in this sense provides us with an early warning system, understanding how the reef is doing, how it's changed in the past, how it's responded previously, and therefore allowing us to understand how it might respond in the future for modeled disturbances. In a nutshell, we can provide that information as scientists, as ecologists, as engineers, and provide that information for managers and policymakers, hoping to hopefully provide information for the right solution. So, for example, if you have a reef where your coral cover has been wiped out because of oil tanker that's stranded and caused a huge spill, once that's been cleaned up and the stressor has been addressed, a probable solution would be restoration because the threat has been removed. Whereas if you have a reef that's been impacted by rising sea surface temperatures, and we have not been able to stop climate change yet, providing restoration in that manner would not be a good fit, but rather you would look at alleviating other stressors that are acting at the same time by providing, for example, a marine protected area. So to simplify, by monitoring, we can provide a diagnosis about the reef, a prognosis, and inform the management actions we need to protect the reef. All right, so having established that it's important to monitor them, what do we actually measure as ecologists when we go out into the field and collect our data? Most predominantly, we're looking at biological metrics. This means, first and foremost, the percent cover of hard coral. So on the seafloor, the benthos, you're looking at the percentage of that is made up of hard coral. What other percent is made of algae, of rock, other substrates, soft corals, sponges, and so forth. Comparing this over time gives us a trajectory of the reef, understanding how coral has gone down or go up, and relate that to different disturbance events to understand what's going on. Secondly, we can go down to a finer resolution, which gives us better information, such as morphology, basically meaning growth form. So looking back at the shapes that I outlined earlier, what kind of shapes are there on the reef? How is that changing? Because each different morphology is associated with different environmental variables it's adapted to. Going down even farther, we can go down to genus, looking at what species there are to ultimately compose species composition maps, understanding how much of the hard coral cover is made up of each species. What does that tell us about the environment? How can we manage that? An example of this is bleaching, prevalence, and severity. So how much of a coral has been bleached? So the response to temperatures that are too high. How is that species specific? How can we better manage that? As well as looking at associated biodiversity. So reefs are not just corals. As I said, they support a quarter of all other marine species. So looking at what kind of fish are there, what is the biomass of the fish, what species, the bioeroders, looking at the sediment around the reef. These are all metrics that we're interested in measuring. Then we go into structural metrics. This means the complexity of the reef. So simplifying it, a size-to-shape ratio. The more complex a coral and a reef in general, the more biodiversity it's likely to sustain, because there's more space for nursery grounds for little fish to go in, and so forth. As well as rugosity. So again, simply put, that's if you look at a reef from its side, look at the 3D structure on top, gives you an indication of how much associated biodiversity it's likely to have, depending on whether or not it's alive or dead. And growth rates, more specifically at a finer scale, again, as it is expected, each species has particular growth rates, respond differently. This changes over time, and understanding that is very important for seeing how reefs are changing now and into the future. And lastly, to put this all into perspective, we always collect environmental variables to explain all of this. So the water quality, things like temperature, nutrient contents, and so forth. All right, so in terms of how we collect this data, I could probably spend hours on this, so I'll just very briefly outline the main ones. We can go all the way up to large scale, such as satellite data that we combine with ground truth in situ collected data to provide habitat maps. Or looking at sea surface temperature from permanent buoys that are deployed in the ocean, as well as using acoustic to look at what different fish there are, because they make different types of sounds, how many fish there are, how healthy is your reef of other biodiversity. But in 10 minutes, I'm just going to focus on the most commonly used survey, which is still the diver or snorkeler survey. And even within that, there are a countless number of different data collection methods, and I'll just focus on the most commonly used one nowadays, which is the photo quadrat. Basically meaning that as a diver or a snorkeler, you have your GPS predetermined transect that you will swim. You'll have a camera look down at the seafloor at a set distance from the seafloor, and every few meters take a 1x1 quadrat photo. All right, and this is where AI comes in, hopefully a little bit more familiar to everybody here. So when you're conducting your photo transect at the end of a field campaign, you might end up with tens of thousands to hundreds of thousands of photos, all of which will need to be annotated. If I were to do that as an expert, that would take me a lot of time, as you can imagine, which is where AI has been amazing because we can upload that. And there's a lot of platforms available nowadays, but using an Australia-specific example, such as Reef Cloud, what it does is it assigns 50 randomized points on your image. You train a small subset of your data as an expert, and then the model will apply that on the rest of your data set. And within a matter of hours, spit out the resolution that you want, depending on the labels you've created. So whether that is just general hard coral cover, morphology, genus, species. And from this, we can construct what we're interested in. So seeing over time how benthic classes are changing at the coarsest level, hard coral over time. And again, we can scale this up if combined with satellite imagery by creating benthic maps. All right, so that's one thing AI is doing really well, but there's a lot of things that could be improved upon in this sphere and beyond that. So most urgently, the need for more accurate and finer thematic scale data. Right now, platforms are really good at being able to identify what is a coral and what is not, but not so great at identifying down to species level. There is a lot of discrepancy because species just look very, very similar, and the amount of training it would require to go down to a very high accuracy is just not feasible to do manually. Real-time inference, so rather than collecting your data, bringing back your data, uploading it, training it, then being able to get your data, having real-time annotation of your data would be a really useful thing to bring in here as well. As the second most common data collection method, 3D mosaics, which Emilio will tell you all about next. We can extract a lot of metrics from this, such as growth rates, rugosity, species ID composition within an actual 3D reef, which is super cool, but at the moment, very time intensive to construct and actually get your metrics from. Then next, predictive modeling. So having that morphology, genus, species-specific information will allow us to much better estimate and model next bleaching events, because right now, a lot of the times it's based on just hard coral cover, and each species responds very differently to stressors. Problem I'm sure all of you are also familiar with is transferable foundation models. So if I were to spend all my time training a model specific to an offshore clearwater reef on the GBR, if I then want to apply it to a second study site on the western coast of Australia, it would perform very poorly. Unfortunately, even if I were to move 50 kilometers inshore from my clearwater reef, where I have a turbid coral reef, it performs very poorly at the moment. Overarching for all of these points is just a general need for less expert dependency. Even annotating the recommended threshold is right now 30% of your data by an expert. If you have hundreds of thousands of photo quadrats, that's still a very painful, lengthy task to do. And it would be amazing to move into a sphere where we don't rely on that as much, as well as having software that's user-friendly, doesn't require lots of coding experience to make it more accessible, and ultimately provide more information to be able to manage areas better. And working together as ecologists and engineers can hopefully tie this all together and move towards keeping roofs looking like this and not like that. Thank you.
Don't go anywhere. We've got a few minutes. Anyone got a question for Faye?
Emilio.
How often can you do like a survey? I know like corals grow really, really slow. So like, what is the frequency such that the survey is actually useful to measure get these metrics?
Yeah, that's a good question. The question is, how often do you survey? for the people online. So typically what we do is at least once a year. Small hemispherical corals, as you might remember from the slide, like the round ones, they can grow only a few millimeters a year. So that won't be captured very readily if you survey more often than that. But branching corals can grow up to between 7 and 14 centimeters a year if they're in an environment where they're happy. So as often as funding allows is usually what happens. But at least once a year, typically twice a year, would be really helpful for looking at how your reef is doing.
I'm hoping you do.
So you said like that the corals look very different depending on in which area you're in.
Yeah.
How do you know as a human? Do you get trained up by someone who's an expert in WA and they tell you that there's corals A, B, C? Or?
So your question is how do you know which corals are on which area of the reef, or which coral looks like what?
Oh.
Okay.
So let's say you go to a new area that you haven't been before.
Yeah.
You know there's like other coral species than the ones that the reefs that you visited. How do you detect those new species? How do you classify?
Yeah, good question. So the question is, keeping both those questions I proposed in mind, like how do you know what to expect when you go to a different site? Basically, if I were to go to a different reef to the Caribbean, for instance, and I have all of my Australia-specific information and training, the species will be different, but a lot of the genera, so the genus and the morphologies will be the same, and at least have overarching characteristics. So we know certain genera are kind of like certain environmental conditions. So if I'm in a reef where there's a lot of wave action, I know what type of genera to expect, what morphologies to expect based on my knowledge from other areas. So there is a lot of overlap that you can generalize depending on environmental variables, as well as where you're on a reef. So on the reef flat, there's usually not as much coral diversity. The corals that are able to grow there are adapted to a lot higher temperatures because the water is shallower and doesn't get as much wash from the open ocean to cool it down. So you get more heat-resistant massive corals, the round ones, rather than branching ones, which like to grow on a steep slope typically.
All right, we might move on, but if we have time at the end, we can always ask more questions as well. Thank you so much, Faye. Emilio.
Oh, right. Does this thing work?
Everyone's doing very well for time, so great work so far, everyone.
Okay. Hi, everybody. For who doesn't know me, I'm Emilio Olivastri. I'm a postdoc here at QCR. And today I'm going to talk about the Bommy Toolkit, which is a project that I've been developing together with my dear friend and colleague, Alejandro Fontan. Ideally, the goal is to enable scalable coral reef monitoring that is easy to access for everybody. So it perfectly joins with what Faye was talking about. It was not planned, but it did work. So how this project was born? It was born because it kind of lied in between my research focus, which is SLAM, simultaneous localization and mapping, which is this problem in which you're trying to estimate the pose of this sensor that you're using to measure the environment, while at the same time building a representation for such environment that you're exploring. In these examples, for example, we have cameras and LiDAR to reconstruct some environments, such as the Colosseum. I am from Italy, so I had to plug it in. When I came here to Australia, I started diving, which is one of the hobbies that I really enjoy to do. And in the picture in top left is me with my friend Alejandro Fontan, which is also a postdoc here at QCR, and the picture was taken by Dorian, another colleague and friend that we will go diving together. So then I asked myself, it started as a hobby, it's like, how can I relive those beautiful moments underwater? So you see all this cool stuff, and you have your GoPro, and you're like, I want a bit more than just looking at the images. I want something that can give me a bit more of context. And then that plus interfacing with people like QCR that actually do coral reef monitoring or more general like just marine monitoring, I started asking myself like, what can I do to preserve this? Just a quick side note, this is not pictures taken from me, it's pictures taken from Dorian. He's a great photographer, I am not. We came up with this pipeline to do photorealistic 3D construction. So ideally, you just need 2 videos in input, and you will have as output a photorealistic reconstruction. So it starts with a calibration video in which you take a video of a known pattern for which you know the geometry properties, and then you feed this video into a process called camera calibration, which will give you the parameters of your camera. If you have a stereo, it will also tell the relative pose between the 2 cameras that will allow you to have metric scale. So that plus the video of the site that you actually want to reconstruct, because otherwise, why go through all the trouble? We run it through some refraction removal algorithms. So what you have is that when you go underwater, you cannot just put your camera just underwater because it's going to break. So what you do is you put it inside of housing. And typically these housings have they're flat. So what happens is that when light passes through water, through acrylic, through air, it gets refracted. And therefore you see like this pattern that is supposed to be squared, it gets bent. The effect is stronger, closer to the edges of the image, and is weaker towards the center. And here we have instead the result of applying the refraction removal to those images, to the image of the example. So then we produce these undistorted images that we can use for our 3D reconstruction process. Ideally, if you're just interested in a specific section that you want to reconstruct, let's say a coral, you can use segmentation networks to produce a mask of just the interest, part of interest that you want to reconstruct. That together, optionally, not necessary, together with the undistorted images are fed through this structure from motion program. It's called ColMap. It's a free one, so that's why we're using it mainly. So to explain it poorly, what it does is ColMap tries to estimate the poses of the cameras and the 3D environment that best explain the pictures that you have taken. So when you feed these images in input, the output is going to be the most likely poses of the cameras that took those pictures and the 3D environment that most explains those images. Which looks a bit more like this. This is actually the sparse reconstruction. I decided to show this one and not the dense one because there are 2 different types. Because here you can see the cameras that are the red rectangles, and the scene that I was trying to reconstruct is that sparse point cloud in the middle. And kind of more or less resembles the trajectory that I was taking underwater when I was trying to do this. But then even the dense one was not enough. So I was like, why can't we just try to plug in those narrow rendering techniques that are able to give photorealistic 3D reconstructions? And from that thing that you saw over there, we are able to obtain something like this, which at the moment looks pretty, and at the moment what it does is just look pretty, but it has so much potential for what it can do afterwards. And here we talk about the Bombing Toolkit in specific, which is this tool that is able to do all those things. It's an open source project, has been developed together between me and my friend Alejandro using Pixie. Pixie is a package manager that allows you to seamlessly install and run these commands. And I decided to hurt myself today and prepare a live demo. So let's see how that one goes. There you go. The zoom is the biggest problem now. There you go. Yeah, there you go. Perfect. So I already pre-prepared some of the things just to make the process as fast as possible. So I collected yesterday night some pictures, few pictures, so it's fast and can be done live, of a scene, and I'm going to show you the different steps. So first of all, for example, if you want to install CallMap, it's really easily. I pre-downloaded the packages, so you're just going to see the installing bit, which we can see is pretty fast, and we have our environment with CallMap installed. And then within Pixie, you can define tasks. So instead of having go through all the steps of the structure for motion pipeline, I pre-prepared tuning parameters and the different order of the programs to run everything smoothly. So I call this task sparse reconstruction, and then I add the path to where I have my images. So hopefully, yeah, you can see that everything runs smoothly. And we have the feature extraction, which is the first part, then we have some matching, and then we have the actual reconstruction. And we can see live the result of what we just did. So I go to the demo space, my CallMap workspace, and I go to my sparse reconstruction, and I show you this. So very sparse, it's just 12 images, take into account that. But you can see the red rectangles that represent the poses, which I can tell you, trust me, it was actually more or less those poses that I took. And then I pre-run the dance one, so you can also see that one. So you can see that we gain a bit of detail. But still, just from those few images, this is the best that CallMap can do by itself. So, and this brings the, hopefully, yeah. He does bring this second bit, which is the photorealistic reconstruction that I'm just going to run now. Yeah, which is gonna be fast because I pre-run it once, ideally. I also have a plan B in case it doesn't work. Because
on HPC?
This is running on HPC because I don't think my PC can handle Zoom and, and all these things together. Okay, well, it is not running, of course, but I can show you. Not this one specifically. Oh, actually, we have it. Oh, okay. Okay, this one is not running at the moment, but I will show you the other ones that actually are underwater scenes. So this one was some data from Malaysia that collected from the marine biology group in our block. Was provided by Brett. And these are like some stars where you connect the corals, and you can see a very great level of detail in these 3D constructions. Then we have another site in Malaysia. Also, we have these cement structures in which they plant coral to make sure that they are able to grow in these stable structures. And finally, we have the, oh, there you go, nice. And finally, we have the reconstruction that I showed the example, which is just beautiful in my opinion. Like, I just keep looking at it. These kind of projects are born from passion, you know, like, so it's just things that make you happy. Yes. So I'm gonna keep going with the presentation, and hopefully by the end, it's gonna pop up what is going on. Oh yeah, there you go. Come on. Okay. Okay. So there you go. So this is the same thing that we were seeing with the dance reconstruction. And look at how much detail we're gaining just by using this kind of representations. This is the 12 images. This is just from 12 images. And like, I actually hoped it showed less because you can see half of the kitchen. But I think still like it's amazing, and you can just use it with a simple command, and the installation process is seamlessly thanks to Pixie. Yes, it's just a peluche that I have on a pillow on top of the table. But look at like the difference of detail, that how much semantics you gain just from using this kind of representations. Okay. Back to this. I'm gonna leave because probably I'm out of time with what is missing. I'm not sharing the screen. Okay, I don't know why I whispered.
Just missing parts.
What is missing is sharing screen.
Yes, and I don't know. Control-Shift-5? No.
No. Where is Zoom? How do
Where
you
is
change
Zoom?
F11?
There you go.
Okay, and then yeah.
Yeah, there you go. Okay. So what is missing? So at the moment, these are actually just very pretty reconstructions. They are not still used for something other than looking at it. So it would be nice to add some metrics. Actually, this is probably going to be my goal in the next coming weeks, is to add the actual metrics used for marine scientists to evaluate the state of the coral or whatever biome you're measuring. Faye was mentioning the different transects in different years. We want to add scene alignment from different transects, so you can actually compare the sizes of the corals that you're interested in. And ideally, by the end, it's going to be just one end-to-end single command that anyone can use, and you will have from videos to neo-rendered representation with potentially some of the metrics that you're interested in. There are so many other things on the list that I could not fit in a slide. But yeah, if you have any questions, feel free to ask. Oh, this is the link. If anyone wants to access the GitHub.
Emilio has time for one quick question.
Okay. Yes.
So on the calibration pipeline,
Yes.
I find that a lot of the trouble I have is we upload images to move the camera too fast.
Yes.
Is there anything in the pipeline that detects this?
Yes, there is. It has been removed at the moment, so it is still there. But we do it, so what we have a video, we get the frames, and then we synchronize those frames. So at the beginning, we have this thing about removing the images that were blurry, but then would mess up the synchronization. So we just revert the 2 steps, and now we have synchronization. Then we remove the blurry images that also impact not only calibration, but also impact the 3D reconstructions, which, yes, so now we do have that too.
Cool. Thank you so much. All right, and our final speaker, Beverley. Really, we need to.
Oh. Yes.
I'll do the slide if you like.
Excellent. Thank you. Thank you. For those who don't know me, I'm Beverley. I'm a PhD researcher here at QCR with Toby, Alex, if he's here, I can't see him, and Michael. So today I'm going to talk about a project we were working on at the beginning of this year called Long-Term Multi-Session 3D Reconstruction Under Substantial Appearance Change. So I feel like it follows on nicely from what Emilio just spoke about. But starting with motivation, Emilio conveniently said that we're missing multi-scene alignment. So our goal is long-term 3D mapping across multiple visits in substantial appearance change environments. And our focus is on 3D mapping because it's a crucial tool for environmental monitoring, ecosystem, like understanding how it changes, and monitoring health management. Coral reefs in particular, I'll press the right button, as Faye also very well described, they are highly valuable, very biodiverse, but are increasingly affected by climate change and other human stresses. So it makes them very important for us to focus on. Oops, I keep clicking the wrong button. Visual survey-based monitoring relies on being able to align different scenes over years. And some examples I've shown here, they look a bit dark on the screen, but we're looking at coral reefs either years or months apart. But this can be really difficult for existing structure for motion methods due to strong change between appearances over years. The coral itself is changing, whether it's growing or degrading, and also issues like light management underwater, depth, robustness in distortion and things like that. So in this work, we focus on trying to reconstruct a single coherent 3D model from temporally separated surveys, and we argue that the main limitation in existing methods is robust cross-session correspondences. So being able to make associations between images from 2016 and 2017, for example. So here I've included an example of the output of our work, and I'm going to build up to how it works, but I just wanted to start by showing that our end result is this aligned set of 3 point clouds that cover images from temporally separated surveys across 3 different years. And we make the argument, like I said before, that the main challenge is establishing correspondences between different visits. So if you're not familiar with structure for motion, you are now, because Emilio just explained it to you. But briefly, I would say that it's a method for reconstructing 3D geometry from a set of 2D images. And it does this by collecting salient key points, so key points that visually mean something. An example might be the edge of an object or change in intensity. And I've marked examples. The edge of the statue's thumb might be an example of a key point. And then it matches descriptors of these points between images, and it does that to estimate the 3D geometry and the camera poses jointly in the optimization. And this can seem relatively straightforward for an example like this, where it's a rigid object, it's photographed under ideal lighting conditions, and all the images are taken at the same time, so nothing's really changing. But this does become more complex in our underwater environment. So like I said before, the coral may be changing and the conditions are more difficult, and when we have things like wildlife getting in the way as well, that can throw a spanner in the works in the system. So let's just take a look firstly at what would happen if we used this naive approach in our kind of multi-reef environment. So what I'm going to describe is we call it joint reconstruction, or this naive approach where we just throw all of the images from 3 different years at once into the system. And what we expect to get is this nice point cloud with points from 3 different years, and the same coral in one point in 3D space should align to that same coral in another year. And if we run it on ColMap, which Emilio just explained, or another method called Map Anything, you can see over on the right that ColMap can only reconstruct 1 year's worth of information, and Map Anything looks like it can produce maybe 2 years worth, but we see it's fragmented, it's disjointed, and we argue that this is because the system, while it can do a good enough job within the same year, it can't make associations between different visits for whatever reason. So clearly this doesn't work in our long-term substantial appearance change kind of scenario. If we try another method, we can try instead reconstructing each year separately and then aligning them in a post hoc manner. And that should at least make sure that we don't lose as much information as before. What we can see here using iterative closest point or a method called BufferX, it does get us what looks like all of the information. This is just the sparse method, but you can kind of get an intuition that something's not quite right. They look closely aligned, but you can see that the camera poses look on different levels. And that tells us, which I'll show in a moment, that at the pixel level, there are some discrepancies. So we're not entirely happy with this one as well. Whereas our method produces a complete, coherent, and aligned 3D point cloud with all 3 years joined together. I'm going to explain technically how this works in a moment, but I just want to narrow in on why this is bad and this is good. So having a look at the pixel level, this visualization shows some input key points from one session and their ground truth partners in another session. And like I showed before, and this is a closer look. The post-hoc methods show some pretty noticeable misalignment at the pixel level, whereas our method achieves almost perfect pixel level alignment. And then on the bottom row, the geometric warping closely agrees with what you would expect if you were to take the exact two images, the points from those two images, and overlay them together. Quantitatively, our method achieves a reprojection error of 3.65 pixels, and this is an improvement of, I think, about 95% over both post-hoc baselines. So I've explained why the other methods don't work. I've shown that our method works. I'm going to conclude by explaining more technically how our method works. So this is an overview of the pipeline. We take, similar to the joint reconstruction method, we take all the images from every session at once and we input them into this feature matching and feature matching pipeline. But what we do as well is we input this similarity matrix, which is computed using visual place recognition. And for those unfamiliar, I won't go into it in too much detail, but VPR is a tool to match the same place or the same image from different sequences. And it does that by computing a similarity between the feature descriptors that I described towards the beginning. So with these two things together, we take the images from the same session and we do a fast handcrafted feature matching exhaustively across all those images from the same year. What we do differently to what I explained in the beginning was we use this similarity matrix to get candidate cross-session pairs, so they're most likely the same place, and we selectively apply a learned feature matcher, which is more robust but a bit slower, only on these candidate matches. We then enforce these matches in the structure for motion optimization. So you might be wondering, why not just do exhaustive learned matching across every visit and every image to get the most tightly coupled reconstruction that you could possibly get? Having a look at computational complexity, we've evaluated 4 different configurations, each becoming more selective in how it does this learned feature matching, with the final one being what I described at the beginning, which we know doesn't work. Restricting the learned matching to only the VPR-based pairs, we see an improvement computationally of 95.5%, while also still achieving the best mean reprojection error. So we're very happy with that. And this also shows that as your data set scales and you get more images in your sequences, it still should be tractable. And I just spoke about scale. So the examples that we showed before were on a 3-meter radius area on the same reef in Okinawa. And what we did to conclude is to increase that to the entire data set, which is a 30-meter radius, and that corresponds to a 60 by 60 meter area, which is quite impressive that we can still get this aligned, coherent, and complete point cloud. We also tried it on another data set from Lizard Island, and this is information from February and March before and after a major bleaching event. So we're happy with these results as well. That's all I have today. Thank you.
Thanks so much, Beverley. We do have time for questions. Any questions for Beverley?
You? Oh, yes.
Does that help with like the banana effect and stuff?
No. So one of my next steps is to eliminate the refraction and the distortion, especially in this one, because there's a fair bit of distortion. This is a top view. The side view is a lot less attractive. So, yeah.
So you briefly mentioned next steps, but is there a bigger picture for the sort of final work of your PhD? Are you hoping to measure change between these times?
Yeah, eventually. I feel like this is my second work in my PhD, and they've both been focused on enabling change detection. But what I'd really like to do is work with ecologists and look at how we actually can do change detection. Yeah.
Sorry. Great. Well, thanks so much, Beverley. And can we thank all of our 4 speakers again as well?
Amazing.
All right, that's it. Finished perfectly on time. Well done. And feel free to keep eating food and hanging out, chatting as well.
Emilio, that thing's so bad.